Hello, we're glad you've joined us for this live webinar, Integration of 3' mRNA-Seq and iCLIP to Study the Regulation of Alternative Polyadenylation. I'm Judy O'Rourke of LabRoots, and I'll be moderating this session. Today's educational web seminar is presented by LabRoots, the leading scientific social networking website and provider of virtual events and webinars advancing scientific collaboration and learning. It's brought to you by Lexagen. Lexagen is a transcriptomics and next-generation sequencing company focusing on the development of technologies for complete transcriptome sequencing. Its portfolio includes innovative molecular biology kits, software, and services for RNA Seek. To learn more, visit www.lexagen.com. Let's get started. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. You can pose questions to the speakers during the presentation while they're fresh in your mind. To do so, simply type them into the Q&A box, which will open when you click the green Q&A button at the lower left of the presentation window. Questions will be answered after the presentation. To enlarge the slide window, click on the screen icon in the lower right-hand corner of the slide window. If you experience technical problems seeing or hearing the presentation, just click on the support button found at the top right of the presentation window, or report your problem by clicking on the green Q&A button at lower left. I now present today's speakers, Gregor Rot, PhD, and Ule, PhD. Gregor Rot obtained his Master of Science in Computer Science from the University of Ljubljana in Slovenia and his PhD in Bioinformatics from the University of Zurich in Switzerland. Gregor is collaborating with several research groups performing data analysis and developing expressRNA.org, an interactive research platform for the study of post-transcriptional modifications. Ule obtained his PhD in molecular neuroscience from the Rockefeller University in New York and started his research group at the MRC Laboratory of Molecular Biology in Cambridge. In 2013, he moved with his group to the Institute of Neurology at University College London, and since 2016, the group is based at the Francis Crick Institute. The group studies how protein RNA complexes regulate gene expression. His group developed iCLIP, a transcriptome-wide method that identifies protein RNA and RNA-RNA interactions in living cells with high resolution. This method is employed in combination with molecular, cell, and computational biology to study how the structure and dynamics of protein RNA complexes regulates neuronal differentiation and function, and how their dysfunction contributes to neurodegenerative diseases such as ALS. The group also studies the role of transposable elements and non-canonical splicing events in the evolution of alternative splicing. Dr. Rot will now begin his presentation.
Well, thank you for the introduction, and welcome to everybody attending. And in this webinar, we'll be talking about 3' end sequencing data to study alternative polyadenylation. And we will look on how to integrate this data with iCLIP data, so RNA protein binding data, to better understand the process of alternative polyadenylation. And we will be doing this by looking at a platform called expressRNA that we developed to integrate all these kinds of analyses together. So, but first, a little bit zoom out on the topic of polyadenylation and cleavage and polyadenylation in general. So if you look at the line here in the middle of the slide that represents the RNA, you see there are many protein complexes binding on top of this around a polyadenylation site, so the point of cleavage. Cleavage factor, cleavage and polyadenylation specificity factor, and so on. So, and you also see that there are specific cis elements on the RNA. For example, the polyadenylation signal, which is a well-preserved hexamer. There is a U-rich region, and there are UG-rich regions and other elements. And this whole cleavage and polyadenylation 3' end processing machinery is then also interacting with other RNA binding proteins. And we are interested in one specific protein. We will be talking about one specific protein, TDP43, which is involved in neurodegenerative disease. And our study is interested of how this protein is disrupting polynylation. So preventing the 3' end processing machinery of doing cleavage and polynylation on a specific site and switching this process to another site, mostly upstream of the gene. So how to study and how to develop software to tackle this problem in a way. So what we did, we developed a platform called expressRNA, and a specific part of this platform, which is intended to study alternative polynylation, is called expressRNA.APA. APA stands for alternative polynylation, and it handles all the aspects of the analysis, from taking the raw sequence data to managing, annotating the data, using state-of-the-art tools to map the reads to the reference genome, then computing differential polynylation analysis, and even going further and combining this with alternative splicing, and you will see later also with motif analysis. So expressRNA is really intended as a post-transcriptional analysis research tool. So back to polynylation and alternative polynylation, how to study these processes. So usually what you do, and in this case the data is coming from Lexogen quant reverse protocol, you prime with an oligo-DT primer, and by doing this you catch only the end of the RNA molecules. So the molecule and the section where the molecule is then polyadenylated. And then you do high throughput sequencing, and not to go into detail, you get millions of reads that are containing the signal of where the RNA was polyadenylated, and then you map these reads back to the reference genome. And for example, in this graphic I'm showing you a distal site or a proximal site, and then you group these reads together. And here on the right you see you do a little bit of clustering, because cleavage and polyadenylation is not a nucleotide exact process. So we have a tolerance of -5 to 5 nucleotides in our case. And after you group these signals together, you can imagine that you have information in a genome-wide manner of where polyadenylation is happening, and not only where, but also the extent of polyadenylation. And the immediate next question is, if you have your own data set, you would like to understand if the polyA sites that you are detecting are valid sites. So if other people already detected this polyA site that we are looking at. And what we do immediately here, we compared our polyadenylation database, if you call it like that, or atlas, to 2 other studies by Doherty and Gruber, and you see that the coverage of the location of the polyA sites is strikingly similar. So there is a little bit of change in a couple of nucleotides, because as I mentioned before, the cleavage is not a nucleotide exact process. So you have to tolerate a little bit of change in this way. But then you do further analysis to validate your polyA sites, and here what we do is we look at nucleotide frequencies, and on the left side you see graphs from our data set that we detected and that are also covered by other data sets, and you see an A enrichment upstream of the cleavage polyadenylation site, which is here in the center. And you also see a T enrichment downstream. And this is really a significant, let's say, pattern of polyadenylation cleavage site that you expect from your data set. And then if you also look at our polyA sites that were detected but not detected by other studies, you see a similar signature, and it's less pronounced, but still this gives you an idea that also the sites that we detected that are novel are also valid polyadenylation sites. And then to further validate this, we developed a de novo motif search algorithm. And what you would expect from this kind of search is that it's also very well studied that the polyadenylation signal is present 20 nucleotides upstream of the cleavage site. And it's very well known also which kind of signals are the most frequent. So it's very well conserved hexamers, AAT, AAA, and variations on this theme. And if you look at all our detected polyA sites here in green, you see the signal is very strong and it's where it's expected to be. And you also, if you look now at the novel polyA sites that we detected with our data set, you also see in this red dotted line that is pretty strikingly overlapping. And this is another, let's say, this is evidence again that the detected polyA sites with our data sets and with our data are true polyA sites. Next, what we did is we took our RNA Seek data sets. So classic RNA Seek from Hexa cells, since also our experiments were in Hexa cells, and we were interested to see if we look at the coverage of the RNA Seek just upstream of the detected polyA sites, if the coverage is dropping. And so here on this graph upstream, you see on the top, you see that in this green line, there is a drop in coverage just before the cleavage site here. And this is true for the polyA sites detected by our and other studies, and also for, to a little bit less extent to novel polyA sites that we detect here in red. And the middle and the bottom heat maps, they show you the top coverage for genes in terms of a single gene. So each line in the heat map is one gene. And again, if you look at the RNA-Seq data, you see a drop in coverage. And if you look at Lexogen QuantSeq data, you also see a striking signal just before the polyadenylation cleavage site. And this again shows you that the data is valid. So we went to through a lot of work to really validate the polyA sites to show that we are working with really significant data. So if you imagine now where you know where the polyA sites are, and so where polyadenylation is happening, and you also know the extent of it, the immediate question is how to detect genes which are alternatively polyadenylated. So where a switch occurs and polyadenylation is switched from a distal site to a proximal site, or vice versa. And in the beginning, we had this simple approach where we computed the percent change, and we looked at ratios between the controls and the test. So the controls are wild-type cells in this example, and the tests are a knockdown of a protein of interest. And we classified genes in three categories. So genes where the proximal site is upregulated and the distal site is downregulated, genes where we have observed no significant change here in the middle, and then also genes where the proximal site is downregulated and the distal site is upregulated, talking in terms of control versus test always. And we only look at two most significantly changed polyA sites per gene. So we decided to do this because if we look at all the sites, it's becomes very, very complex. And also in terms of biology, you always would expect to have a very significant change, a switch from one site to one or two other sites maximum. And how do we do it currently with expressRNA? So we use a software called DexSeq, which is state-of-the-art in terms of looking at alternative splicing, but instead of providing counts for exons, we provide counts for polyA sites, and then this software tells us which polyA sites are significantly changed, and we select the two most significantly changed sites, and we also require that they change in the opposite direction. And by doing this, we can really classify each gene for which we detected at least two or more polyadenylation sites into one of these three categories that I've been showing you in the previous slide. And we do an additional categorization. So based on where the selected proximal and distal sites are in the gene, we classified the genes additionally into this same exon, composite exon, and skipped exon category. And for example, the same exon simply means that the proximal and the distal sites are in the same exon. It doesn't need to be in the last exon, it can be in an exon somewhere upstream. And the composite exon and skipped exon scenarios are a little bit more complicated, so we also look at splice sites around these sites, so this 5' splice site and 3' splice site, because we were also interested, it will be later evident in the so-called RNA maps, where we look at the binding of a protein to the RNA. We were also interested if a protein is regulating polynylation and splicing at the same time by binding around the polynylation sites and also at the 5' and 3' splice sites. And I would like now to pass the presentation to Jernij.
Okay, so I'll be happy to take over now. So the next question we wanted to understand is whether the polyA sites that are identified by QuantSeq are directly regulated by specific RNA binding proteins. And really the goal of our lab is not to identify polyA sites, and probably there are better methods than QuantSeq to identify new polyA sites. We are rather interested in understanding which sites are regulated and what proportion of those are regulated by direct binding around the polyA sites, which ones are regulated due to the splicing changes, as Gregor has just described, and how this combinatorics works when different proteins assemble on the same RNA. And for this purpose, we actually need to integrate a variety of different data sets. So on one hand, Gregor described how this regulatory function can be identified through the QuantSeq, which can be through alternative polynylation. We can also input into the pipeline that he developed, the expressRNA, the information about alternative exons that can be identified through standard RNA Seek methods. And then we want to understand the different type of interactome data that are enriched around those sites. So one method that we use in the lab a lot is the iCLIP method, and on the other hand, Gregor also works with tools that can directly discover regulatory motifs. So now we have the information about binding, information about regulation, and we put them together in different kinds of ways. And one way that we do this for just kind of visualization purposes is to draw this what we call an RNA map, where we are looking at the positions where protein binds to either enhance or repress the use of certain polyA sites. So as I described, one method we use is the iCLIP. This is a biochemical method to define the sites of interactions between proteins and RNAs. And one of the kind of principal steps of the method is that you can cross-link these complexes in vivo, so the interactions are preserved and representing the in vivo environment. We fragment the RNA to pretty short pieces, and that ensures that we will not get a lot of indirect interactions. We will mainly get contacts of our protein directly to the RNA, rather than purifying large complexes of lots of interacting partners. During our purification, we ligate an adapter to the RNA, and this adapter will be complementary to the later primers used for reverse transcription. And in order to be really stringent with our method, we tend to visualize our complexes on an SDS page gel. So we can, first of all, see that we are having only the complex of interest present and that the controls are clean. And secondly, it allows a further purification of the complex. And then we prepare the cDNA library. The central aspect of the iCLIP protocol is that it can actually amplify the cDNA that truncate at the point of cross-linking. It only allows one adapter to be present on the RNA, and it can amplify these cDNAs by introducing the second adapter after the point of reverse transcription. And once you sequence this data, the analysis actually relies on looking at the nucleotide preceding the mapped reads. So the nucleotide on the genome that precedes the map read is where we expect most cross-link events will locate. So that's the analysis of binding interactions through the experiments. And you can look here a comparison of binding analysis that is either based on the sequence. So we know that this protein that we study, called TDP43, likes to bind UG-rich sequences. And in this particular RNA, you can see I just put an arrow next to the site in the intron where the protein has a strong UG-rich sequence, and then another arrow in the 3' UTR. And you can see the structure of the gene here underneath. So the bigger blocked regions are the exons, and then the arrowed thinner line are the introns. And here we have an alternative exon that is being regulated in red, and TDP43 promotes inclusion of this exon. And you can see that there are binding events nearby near the exon, and that the regions of UG-rich sequence tends to also overlap with regions of strong cross-linking. And generally, we see there is a close correlation between the binding motifs of a protein and the cross-linking signature of the protein. So next, we can understand how this correlation between binding and function really looks in a more systematic fashion across the larger set of regulated exons. And this is where we come to this point of looking at the data in the form of an RNA map. So on one hand, I just noted before that there is a close correlation between enrichment of UG-rich sequences and strong cross-linking of the protein. And on the other hand, we can see that the positions where the protein cross-links depend on the function. So here you can see a group of RNAs which have strong cross-linking of the protein just upstream of the exon. And the exons in this case are all the repressed exons. So that's why we see these blue dots present upstream of the exon. Similarly, we will have exons that will have enrichment over broad regions downstream of the exon. Again, those are repressed exons. So whenever we make a blue line, it means that this is an exon that will be skipped in the presence of TDP43. And then we see a last category of exons which has binding just downstream of the exons, and these exons tend to be enhanced by TDP43. So very often you see that the same protein can have dual functions. It can either enhance or repress splicing. And here in this study, we wanted to understand, is it possible that some proteins will have even further functions? Not only they can regulate splicing, they can also regulate triggermen processing. And here is now the RNA map of triggermen processing. And it's a little bit complex, so I'll explain it step by step. So first, we can see it in the same format as we did it for splicing, where we just plot every single RNA in its own line. And you can see generally that binding of TDP43 is quite enriched just around the site of cleavage, and also close to the site where the polyA signal is located, which indicates that TDP43 will tend to block these sites and hide them from recognition, which is kind of consistent with the fact that it's repressing these sites. And you can see the same type of a map in a summarized format, where we are now not plotting every individual RNA, but we are summarizing data across all of the regulated sites. And this map also involves some statistical analysis. So first of all, I should point your attention to this central gray region in the middle of the map, which represents binding on RNAs that are not regulated. This would be what we would consider control RNAs. So it's good to have some information about how the protein binding distributes in general around the polyA sites. And that gives us then an idea whether the regulated sites are any different from any random sites. And indeed, they are different. So we see that when the protein tends to repress triggermen processing, you see these regions of blue color. So all the repressed sites are plotted on the bottom, underneath the control sites. And these regions of filled blue region are regions of enrichment that we can be very confident about. because the blue line here, the one that I'm just going to plot an arrow to, the blue thin line is just the summarized binding across those RNAs. But then you have this gray region around them that represents standard deviation of 10,000 bootstrap analysis of this binding signal. So it's telling you how variable is this signal around kind of summarized binding. So the filled blue region is really taking into account the variance of data as well as coverage. So the regions of field enrichment is really what we would consider significant and reliable. And you can see that the regions of binding where the protein tends to promote inclusion or promote recognition of processing tend to be further away from the polyA sites. And right at the polyA site, the protein will not bind when it promotes recognition of the polyA site. So that's also reassuring that the binding of the protein depends on its function. And you can see the same now in this new slide, where we just plot the top 20 sites which have most binding around repressed polyA sites or enhanced polyA sites. And you see this zone of exclusion for the enhanced sites, which generally will not bind just next to this cleavage event. It will bind further away. So that's really what we are interested in our group, to understand these positional patterns of regulation. And here you can see one example gene. You can see how TDP43 binds very strongly to one specific region in the pre-mRNA. And this region is right next to the proximal polyA site. So the terminology we'll use is the one that creates a shorter isoform is the proximal site, and the distal site creates the longer isoform. And you can see the zoom in into this proximal region, and the polyA signal of the proximal sequence is close to where the TDP43 cross-linking lies. And you can also see how the cross-linking signal nicely overlaps with the regions where we see UG-rich sequences. And the experiment we did here was just to take this pre-mRNA sequence out of its context, put it into a reporter assay, and then measure the ratio between proximal and distal signals in the wild-type cells or knockdown cells. And you can see that in the knockdown cells, we are now making more of the proximal isoform. Normally, TDP binds there and represses processing, but when you take it away, we are making more of the shorter isoform. And when we mutate the TDP43 binding, then the difference between wild-type and knockdown is lost. And actually, already in the control cells, we will make more of the proximal isoform, because now we're not inhibiting this site anymore. And finally, we can actually see that this function of TDP43 can be replaced by another protein. So now we replaced the UG-rich sequences by U-rich sequences that tend to be bound by the TIA proteins. And indeed, we now see that once we do this replacement, then the TIA knockdown will promote the production of the proximal isoform, and TDP43 does not regulate this site anymore. So it seems that this regulation of polyA sites has similar type of regulatory positions regardless of which protein we are looking at, though this needs to be explored more deeply now for larger numbers of proteins. Now, one way that we can also approach this problem is not looking at the CLIP data, but directly looking at the sequences of RNAs. And we've noticed that a lot of RNA binding proteins tend to recognize very short motifs on RNA. Sometimes they are made only of 2-3 nucleotides, and they can even be degenerate sequences. So it's quite difficult to predict the binding sites of RNA binding proteins. But one way we try to get around this problem of short binding sites is that lots of RNA binding proteins have multiple domains. Often they also interact with each other and assemble in larger kind of complex, in this multivalent type of recognition. And one way that one can try to discover these sites then is to start by looking at short sequences. So we can start by looking at trimers, tetramers, pentamers, relatively short motifs, find the first motif that is enriched, and then look, is there any further motif that is also additionally enriched when the first motif is enriched? So we're now looking at coupled enrichment of multiple motifs. And Gregor can explain more later on if you're interested about how specifically this statistical analysis works. But just to give you how this works in case of the polyA sites regulated by TDP43, if we look at the polyA sites that are proximal, we see that when these sites are repressed, then the motif that is mostly enriched in this first region upstream of the polyA signal is the GUG motif. And actually the same also happens at the position that overlaps with the polyA signal and the cleavage side. And interestingly, when we plot this motif around the two types of sites, enriched or enhanced, we see that the region downstream of the cleavage side will have enrichment at the enhanced side. So again, consistent with what we've seen with the iCLIP data. But the advantage of this motif analysis is that sometimes it can also uncover other types of motifs that actually you would not expect to be enriched. They are not known to bind TDP43. They are enriched in this case downstream of the repressed sites. That indicates maybe other proteins binding at those regions, and somehow these proteins might be interacting with TDP43 or competing, and kind of opens up new opportunities for setting up new hypotheses. And again, in this downstream region, the software also detected enhancement of this UG-rich motif. So basically, this position-dependent search for cluster motifs is quite efficient in uncovering regulatory motifs. And it also shows that the distal sites can also be directly regulated. So for example, it can discover the UG-rich motif also around the distal site, showing that, for example, TDP43 can repress the use of distal sites if it binds nearby of these sites. So it really shows how there's a lot of different ways how TDP43 can regulate pre-mRNA processing. So just to summarize this diversity of regulation, here actually I was just showing you data for the tandem sites. So as Gregor described earlier, the tandem sites are the ones that are present in the same last exon. We have lots of cases of repression by binding just next of the cleavage site, enhancement where you're binding further away. We see that TDP can also directly regulate distal sites. And what I haven't shown you is that we also see regulation of other types of polyA sites, for example here, the skipped events, so the ones where you have actually a splice site in between and you're on different types of exons. We again see this, for example, enrichment close to proximal sites that are being repressed. So just to wrap up, I'll pass it back to Gregor.
Okay, thank you, René. So just a final slide in terms of availability of software in terms of analysis. So expressRNA, it's an online web application when for now you can explore the results that we were talking about here. And it's composed of 3 different Python modules. So this APA module, which is taking care of the pre-mRNA sequencing data and the processing. And in this module integrates several external state-of-the-art tools for mapping to the reference genome, doing various analyses and so on. So we really took different software and tried to combine it together in a way to serve our purpose, our research goal. The RNA motifs 2 is a completely independent Python module which is used to do the motif analysis that René was talking about in the previous slides, which you can download and use completely independently, but we integrated it in expressRNA. And PyBio, it's a small tool set of genome annotation tools. So it facilitates the download of genome annotation, for example, from Ensemble. And if you have a genomic position and you would like to know which gene it's annotated to, or if it's intergenic, so you can do things like that with PyBio. And now I would like to describe a little bit a video that we will play for the end, which I cannot comment live, but this video will just show you how to use expressRNA online and how to explore it. You will see that there is a help section where we describe the basic concepts, and I think in the end you will also see a RNA Pac-Man game, which is still in development, but I think it will be a lot of fun. So we'd like to ask now the moderators to play the video, please. So we come to the last slide. I would like to thank Tomasz from the University of Ljubljana and Mark and Christian from the University of Zurich for all the help with bioinformatics. Then Ina, who is now a postdoc in Heidelberg, for doing the experimental work for the data that we were showing. And Micha, who is finishing a PhD in Munich, also for all the help and ideas. And Jen, who is now working for a startup in Paris, and she did a lot of work in developing pre-mRNA sequencing protocols. And Ina, Micha, and Jen were all part of Jerne's lab here on the picture. And I would also like to thank Jerne, who was really instrumental in designing the experiments, guiding, and also with a lot of ideas and help with bioinformatics and motif analysis. And I think we can now move on with the question and answers.
Thank you,
I'd like
Dr.
to thank you, Gregor.
Great. Thank you both, Dr. Rot and Dr. Ule, for your presentations. A quick reminder for our audience on how to submit questions. Simply type them into the Q&A box found by clicking on the green Q&A button at the lower left of the presentation window. Dr. Rot and Dr. Ule will answer as many questions as time permits. And first question is for you, Dr. Ule. How long is the usual polyA signal, and is it over 3 consecutive A's or more?
So it's a hexanucleotide. The most common signal is AAU-AAA. So you can see in one of the slides we showed the, I think, 12 most common hexanucleotides. Definitely AAU-AA is the most common one. It's maybe present at about 40 to 50% of the sites, I believe. Gregor can correct me. But this is a well-studied element that has been characterized a lot by other studies already. So we just use it as a quality control. And it binds to the CPSF component of the processing machinery.
Okay, here's our next question, also for Dr. Ule. You showed slides by Ron et al. 2017. Was that picture published?
Right, okay, just a moment. So that was slide 7, if I understand right. I think you probably are referring Rot et al. Maybe that was a misspelling, Ron et al. And I'm pretty sure that it was published. What we have shown, I believe, has been all published. So it may be in the supplementary material of our paper, I'm pretty certain, if I'm looking at the right slide.
Okay. Here's our next question. Person says, hi, thanks for the presentation. I have a question. How much is minimum coverage of sequencing for APA discovery?
So the coverage is difficult to estimate. If you have around a couple of million reads in an experiment, you can already start to draw some conclusions from the experiment. Because especially it's interesting because since you don't sequence the entire transcriptome, but you are targeting the end of the molecules, the RNA molecules, you need a lower coverage, I would say.
The next question is, can I use expressRNA for non-model organisms with genomes not well annotated?
So thank you for the question. Yes, it's possible. So expressRNA is built such in a modular way that you can add external genomes. For example, it's quite easy to add genomes that are, for example, present in ensemble. So any genome that is available there, you can add to expressRNA.
And the next one is for Dr. Rot. How do you deal with internal priming events in the mRNA-3' end sequencing approach? And do you have filtering approaches in expressRNA.up?
So yes, we are aware that internal priming is a big problem in the field, and what we do is we do two things. So first we look at the genomic sequence surrounding the polynylation cleavage sites, and we filter out with certain thresholds of enrichment. And the next thing we do, we use a classification software called polyA, and this software classifies, again, it looks at the sequence, and it was trained on a set of sequences of polyA sites, and then this software is able to classify a specific polyA site into a strong, weak, or a site without a polynylation signal. And for a very strict filtering, we only look at at strong sites. So we really, for some analysis, wanted to avoid to look at possible internal priming artifacts.
Thank you. Which is or could be the most proper model species to analyze motif complexity involved in the polyA regulation? Yeast, perhaps? I pose that to whomever chooses to answer that one.
So maybe I can help here a little bit, Jarne here. I would say in yeast, generally splice sites and polyA sites are much better defined, so the canonical motifs tend to be stronger, and there are less regulatory motifs involved. So as we go higher into mammals, the regulation becomes more complex. So I do think that in order to model the regulatory motifs, you need to use the same species that you will then use to discover the motifs. So, but I think also it's good to have independent data to build a model. So I think the CLIP data and the fact that we all kinds of CLIP data are becoming available publicly will be quite useful because they get direct information of the protein actually binding, and once we have enough proteins, we can start building kind of combinatoric models, and then that helps to go back to sequence analysis.
This one would be for Dr. Rot. Does expressRNA analyze both QuantSeq forward and reverse data?
Yeah, so we crafted the research platform in such a way that it can take different kinds of pre-mRNA sequencing data depending on the protocol. And it's quite easy, it's just a switch actually to say which type of data it is in a way, because there are small differences in terms of where do you group the reads after they are mapped to the reference genome, either by 5' or 3', or if you have to reverse the strand and other specific technicalities like this. So yeah, it can handle both types of data, and also not only QuantSeq but also other types of data.
How do you define the PA cleavage site for mRNA?
So the cleavage site we define it by looking at the data, and then we look at specific cis elements surrounding it, and then we construct the polynylation database, which contains all the cleavage sites that we detected in the data.
I guess I should add to that, Gregor. In this case, when we use QuantSeq data, we define the cleavage site based on the QuantSeq data. So it depends whether it's forward or reverse kit. So in case of the reverse kit, it's actually the first nucleotide you get when you start sequencing. That will be the position of the cleavage site. In case of the forward kit, the algorithm that Gregor uses relies on the reads that go into the polyA tail. The proportion of those reads tends to be quite high. So you have to trim those reads, and after the trimming, the last nucleotide of that read will be the cleavage site. And it's a composite analysis once you get enough reads that you can be confident and they survive the filtering, then you can assign that as a cleavage site. Did I describe that correct, Gregor?
Yeah, it's correct.
Okay.
Okay, Dr. Rot, the next one's for you. Do I need to pre-process my data before using expressRNA?
So the data itself doesn't need any pre-processing. Like René was describing, depending on the data, we do trimming and other processing of the really raw reads. So the input that you need for expressRNA are FASTQ files, for example, with millions of sequences.
Okay. Next one is also for you. How do I analyze my QuantSeq data via expressRNA?
So for now, the online application doesn't allow users to upload their own data, for example, to be analyzed on our servers, but that's in plan, let's say. So the way to go would be to download the Python packages, so APA especially, and set it up on your server or your desktop computer, since it's not so CPU demanding, and then annotate your data and do the analysis with it. And there are some dependencies that you also would need to install, like if you want to use a specific aligner, for example star or bow tie, you would need to install it separately and tell APA where it is located, but these are then technicalities.
Okay, Dr. Rot, next one for you. Can we run expressRNA on our server?
So exactly, this was also connected with the previous question, and yes, you download the Python software, you set it up, and then you can run it on a server or on your desktop computer.
Okay, and next one is for you too. How can I use expressRNA to identify motifs enriched around a predefined list of alternative exons or polyA sites?
So in this case, if you have a list of exons or polyA sites that you would like to search for enriched motifs, I would suggest to download the RNA motifs 2 package, which is also a Python software. And then you can look how to provide this list into this software and just run the analysis on your computer. It doesn't need to be integrated with expressRNA. This motif analysis software is completely independent. We just integrated it with the online web application since it's very convenient to run the analysis on the data.
I would like to once again thank Dr. Rot and Dr. Ule for their presentations. Do you have any final comments for us today? Dr. Rot?
Yeah, thank you for the opportunity, and I'm very happy to present our work.
And Dr. Ule, would you like to share some thoughts? Okay, we will close. I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today will be addressed by the speakers by email. We'd also like to thank our sponsor, Lexogen, for underwriting today's educational webcast. This webinar has been approved for continuing educational credits. Please click on the CE button at the bottom left corner and follow the process to receive your credits. This webcast can be viewed on demand through December 28, 2017. LabRoots will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.